everyone. In this video, we will describe channel-mediated diffusion. There are several different types of channels that are involved in channel-mediated diffusion, so we'll just label them quickly. They are leak channels, ligand-gated ion channels, and voltage-gated ion channels. And aquaporins are used for water transport, so we won't consider them as part of channel-mediated diffusion because technically they get their own category, which is osmosis, and so therefore they get a separate video. But I do want to mention that the aquaporins are simply protein channels. It's worth just sort of mentioning that just to sort of keep everything into context. So what we're going to do is describe in detail each type of channel and how the diffusion process would work for the different channels. But first I just want to mention that the channels are specific. So we could have, for example, a potassium leak channel. We could have a sodium leak channel. And when we talk about the different types of leak channels, we'll usually label them by which ion is going to move through them. And the ion is the only ion that's going to move through them in most cases. So as long as there is potassium available to move through the potassium leak channel, we would never see sodium moving through the potassium leak channel. But every now and then, if there was absolutely no potassium to be found anywhere, is it possible that a sodium could squeeze through? Maybe. But I think it's really it's just easier to think of them as specific. And most of the times that we'll talk about them, we're just going to sort of say, yes, there's leak channels present, or no, there aren't. And I think it's better to just sort of know that that it's specific for a certain ion. Ligand-gated ion channels are similar. We could have a sodium ligand-gated ion channel. We could have a chloride ligand-gated ion channel, and those aren't the only ones, but those are the examples we'll cite for right now. And then similarly, voltage-gated ion channels are also specific. Sodium voltage-gated ion channel, potassium voltage-gated ion channel, calcium voltage-gated ion channel, and so on. What we're going to do in this video is pick an example of each type, leak, ligand, and voltage-gated channels, and describe what happens in terms of the movement of the ions. But I just mentioned that there's multiple examples because I don't want to give the impression that there's only such a thing as a potassium leak channel, but there aren't other types. There are other types, and it's just that we're just going to label them by which ion can move through. We will draw a cell membrane. We'll label the ECF and ICF, and then we're going to draw in some ions. So potassium, as an example, is highly concentrated inside of the cell. So we'll show that we have a lot of potassium inside and we have only a little bit of potassium outside. So high concentration potassium in, low concentration of potassium out. The brackets represent concentration. So putting the brackets around the K plus represents concentration. So because we have high concentration of potassium inside the cell, low concentration of potassium outside the cell, we know that a gradient exists between these ions. There's a little bit to say about the charges as well, but for this moment, I just want to sort of talk about the concentration of the actual ion, and then later on, when we start talking about transmembrane potential, we'll talk about the charges and the impact the charges have as well. Because a gradient exists between the potassium ions inside the cell and outside the cell, we know that the potassium wants to move to eliminate the gradient. Given the opportunity, it will. However, potassium, unlike the other molecules that can participate in simple diffusion, cannot pass through the phospholipid bilayer. It's charged and it's more water soluble than anything else, so it's not able to pass through that lipid membrane. So if we wanted the potassium to be able to move from the inside of the cell to the outside of the cell, we need to place a channel in the membrane that has similar enough chemical properties to the potassium that the potassium is able to slide through that channel. So the leak channel is very much a tunnel, and the tunnel or the channel will allow the potassium to move through the membrane. So the first question is, why does the potassium want to move? And the answer is because it wants to eliminate the gradient. So the reason the potassium moves through the membrane is to eliminate the gradient. Why do we need a channel? Because potassium is charged and water soluble. It otherwise cannot pass through the phospholipid bilayer. So this is very interesting because when we talk about having having the cell membrane be a selective barrier, the membrane decides what things it wants to be able to allow through the membrane and what things it doesn't. And so what the body does is if it wants a particular ion or a particular molecule to be able to move through the membrane of a particular cell, it will plant a channel in the membrane. So if there are no leak channels in the membrane, then there's no possibility for this potassium to move. But if the body wants this movement to be possible, it will manufacture the channel, and then it will stick the channel in the membrane, and then the stuff that is not able to move through the phospholipid bilayer suddenly can move through the membrane, which is kind of amazing. And what's even more amazing than that is that the body actually can adjust what channels are in the membrane and what other types of transporters are in the membrane at any given time. So it could decide, for this reason, we need to have a bunch of transporters in the membrane, and then, whatever has gone on, the situation has changed in the body, and the body doesn't need them anymore, and so we'll go through and take them out. So it's kind of a dynamic process, which is unbelievably fascinating. But for now, just really focus on the fact that the presence of the channel is what allows the membrane to be a selective barrier. It puts the channel there because it wants the movement, or it takes the channel out because it doesn't. The leak channels are always open. So when the leak channel is present, as long as there's a gradient, the ions will be able to move through and try and eliminate that gradient. Now let's move on and talk about ligand-gated ion channels. So the major difference between a ligand-gated ion channel and a leak channel is a ligand-gated channel is not always open. There has to be specific circumstances present in order for that channel to open and allow the passage of ions. So let's again draw a cell membrane, and in the cell membrane we'll draw a channel. And notice that the drawing of the channel is a little bit different than the drawing of the leak channel. And I just do this because it actually makes it a little bit easier to show the closed versus open states of the channel. But it's still a channel protein. That hasn't changed. So we notice also that this channel has a receptor that's present. So we've got a receptor protein that is attached to or part of this channel. Label the ECF and ICF. And let's just pretend that this is a chloride ligand-gated ion channel. So we'll focus on chloride ions. In the body, there is a high concentration of chloride ions outside of the cell. There is a low concentration of chloride ions inside the cell, so a concentration gradient exists between the chloride ions inside the cell and outside the cell. The other thing that we're going to need for this channel to work is a ligand. So we'll draw the ligand, and we'll pretend that the ligand in this case is GABA. So initially, we see that the channel is closed, and if a chloride ion tries to move through the channel, it can't move. And also, the chloride ion is a charged molecule, so it can't pass through the phospholipid bilayer by way of simple diffusion. So the chloride ions can't go anywhere at this point in time. But if we have ligand binding, then the channel will open. So the ligand is going to attach to the receptor that's present on the channel. That's specific, so that would probably be a GABA receptor for the GABA molecule or the GABA ligand. And once that ligand is bound, the channel opens, and then we can see the chloride ions pass through the membrane. So again, the first question is, why does chloride move through the channel? And the reason is because it wants to eliminate the gradient. And the second question is, why does it need a channel? Because it's charged and water soluble, it can't pass through the phospholipid bilayer. The third question in this case that we want to ask is, why does the channel open? And that's because the correct ligand has bound to the receptor that's attached to that particular channel. When we think of ligand-gated ion channels, we can think of the ligand being specific, so we need a specific ligand to bind to the specific receptor, and a specific ion will be able to move through that type of channel or that particular channel. So there's a lot of specificity that comes into play in order for this process to work. But in the same way that we have leak channels, there are going to be certain cells that are going to have specific types of ligand-gated ion channels with specific ligands that are going to interact with those on a very regular basis. Lastly, let's consider the voltage-gated ion channel. So we'll draw the cell membrane again, label the ACF and ICF, and draw the voltage-gated channel into the membrane. Notice that this channel looks different, has more of a boxy appearance, and actually the different types of channels, like sodium versus potassium, have slightly different structures as well, so we'll draw even those a little bit differently. But for now, we'll just identify this as a voltage-gated ion channel, and we'll say that it's specific for sodium. Sodium is highly concentrated outside the cell, low concentration inside of the cell. Like the other ions we previously considered, sodium can't pass through the phospholipid bilayer because it's charged, and if it tries to move through the channel, we notice that the channel is actually closed. What actually causes the voltage-gated ion channels to open is a change in voltage near the channel. So when we say voltage, we're talking about charges. In this case, we're going to show positive charges. So we get enough positive charges in the area of that particular ion channel, it will actually cause the channel to open. We know that high concentration of sodium is outside, low concentration is inside. The sodium wants to move down its concentration gradient, so it moves into the cell. 1, why does sodium move through the channel? To eliminate the gradient. Two, why can't sodium diffuse through the membrane? Because it's charged and water soluble. And 3, why does the voltage-gated channel open? Because of change in voltage, charges near the channel. So in terms of the mechanics of the channel opening and what else is happening inside the cell in order for the channel to open, that actually is going to be explained when we cover something called action potential, which we'll talk about with the nervous system. But I just wanted to introduce the channels now because I think that the more time that's allotted for this to sort of settle in, the easier it will be to become comfortable talking about those other physiologic processes that are happening. Okay, so that concludes our overview of channel-mediated diffusion. As I mentioned, aquaporins are examples of channels, but they have they work differently because they involve water. So we cover those sort of separately in another video. So for this, try and differentiate and keep in mind the differences between the leak channel, the ligand-gated ion channel, and the voltage-gated ion channel. And overall, really think about why the ions move. Understand concentration gradient and understand that the ions move to eliminate the gradient, because that is what makes it diffusion, and that's sort of the overarching principle here that's being utilized. All right, I hope you enjoyed the video. Thanks for watching.